Gene mapping is determining the location as well as the distances among the genes on the chromosomes. There are two types of gene mappings. One is genetical mapping and the other one is physical mapping. So genetic map distances are actually based on the genetic linkage information, which is measured in centimorgan scale. So we get the recombination frequencies among the genes, and from which we get this centimorgan distances, and we put those the genes on the chromosomes on the basis of the distances. Whereas in physical mapping, we actually use the actual distance in terms of the base pairs among the different genes. Synteny is the arrangement of genes on the chromosomes, and comparing syntenies or the gene order between different species tells us about the distances among different organisms. Species diverge from common ancestors. They are supposed to have similar syntenies or gene orders, whereas we believe that gene duplication events, they actually change the gene orders, and as a result species, they diverge. So in this example, we demonstrate the synteny or the arrangement of the genes on the chromosomes. Here we see there is an ancestral species. There is just one chromosome. We have three genes, A, B and C, and it diverges into two species, species A and species B. We see that A has got the genes in the same order, whereas in the B we have seen that A and C, they have kind of flipped their location. So we can have a situation like this or like this, so where we have the same order as we have in the ancestral species, where we can see the synteny is conserved. We see that the metabolically related genes, the genes which are controlling the same metabolic functions, we observed they are found in having clusters which are found together. So for example, here we see the tryptophan synthesis related genes. So the group of genes which are related to tryptophan synthesis, they are placed here in different organisms. On the top is E. coli, then we have Haemophilus influenzae, we have Heliobacter pylori and Bacillus. So mostly these are all tryptophan related genes, they are found together. So we believe their synteny is also conserved. Here we see the human chromosomes. We exhibit them in different colors. Actually, I've taken this figure from Mount's figure 10.7. There is a technique where we break those chromosomes. We color those chromosomes actually, and then we break those segments and put them on some other organism's chromosomes. So in this way, these similar regions, they hybridize with one another, and while looking into the color patterns, we can see which region is among two species is conserved and which part of which chromosome is present while we compare in the second species. So let's take another example. There is a technique called as chromosome painting. What we do here is we allow the chromosomes to react with different staining dyes, and as a result they get different colors. So here we have all 24 chromosomes in humans while we are separating X and Y from one another. So total we have 24 chromosomes here, and we see 24 different colors here. What we do next is we will break them and we will allow them to react with some other the chromosomes from mouse. So here in this diagram we see these actually the mouse chromosomes, and then the segments which are coming from the humans, since they get the similarities between them, so they get interaction and then they get hybridized, and as a result we can see that mouse chromosomes are kind of painted. So here we have, for example, chromosome number 1 of mouse that has been showing us that it is acquiring the segments from chromosome number 1 of human, 18, then we have 2 and 6, and same way we have all other mouse chromosomes painted with the different segments coming from the humans. So they do have lot of similarities with one another, though the arrangement of the genes or order of the genes is not highly conserved. The genes are arranged in different orders, and they are arranged on, they have the segments from different chromosomes of human, and they are arranged on different chromosomes of mouse. So while we do this analysis computationally, we try to make it simplified. So for example, here we have the sequence alignment where we have the sequences which are aligned from one another in the top diagram, and same way these similar sequences, when we put them together in a genome context, will give us the genome alignments. but we see that different regions coming from the genome, they kind of arranged in different orders. So here in this diagram we can see that lot of cross lines coming here from here, and we might have some pictures which are quite messy. For example, here in this diagram, so we can see lot of lines crossing. So actually there are different segments which are conserved between these two species, but their orders are kind of different. So how can we solve these things on the computers? Basically there is a technique in which we simplify that. So what we do is we can take those complex alignments and we can reduce them. So for example, here these alignments have been reduced and they have been simplified. So for example, we have a subsection from the previous alignment where we have 1, 2, 3, 4 and 5, and in the second species the order is 3, 5, 2, 4 and 1. So what we can do here is we can simply invert 5 and 2. So if we invert them, we can have a simplified shape like this. So we can invert, we can recombine different regions, and as a result we can simplify our alignments. So here in this for example case, after this inversion, we can further combine 2 and 3 with one another, right? So 2 and 3 is here. Same way we can combine 4 and 5, and we can further simplify this like this. So basically this simplification is kind of a computationally expensive job, and it needs a good expertise. We will talk about that in some later lectures, but here it was just for introduction. So we conclude that species diverge from common ancestors. They are expected to have similar syntenies, and functionally related genes, they stay close as to one another as a shape of the gene clusters.